Hello. Very good afternoon to the students who have joined. Can you please let me know in the chat box if my voice is audible to you? And also, please let me know if you can see the video and the screen and the presentation. Let me know if my voice is audible to you, and also please let me know if the presentation is visible. Okay, thank you for confirming. We will wait till it's 4 p.m. Then we will start today's session. Okay. Please wait. We will start our summary session from 4 p.m. Till then, if you have any doubt or query, please let me know. It is our last class today, so if you have any doubt or any query, please let me know. Okay. So I hope my voice is audible to you. I will be starting the session at 4 p.m. But before that, I would like to say a few things. That today it is our last class. Okay, today it is our summary session. So in this NPTEL course of Organic Chemistry in Biology and Drug Development, we have covered many topics. Like we have started with our so it was a 12-week course where we started with the basics of biological chemistry and organic chemistry, the interface. We have discussed about molecules. We have discussed about biology, various biomolecules, amino acids. We started with, then we moved with the protein, and we discussed about the various structures of proteins like the primary, secondary, tertiary structures. We have learned how to do the structure determination and how to purify the proteins by various techniques of chromatography. Then we move toward the kinetics and enzyme kinetic studies, the different kind of kinetics, the modes of concepts of enzyme inhibition we have discussed. Then we discussed the nucleic acids, the DNA, the sequencing method, followed by the central dogma, which is the core principle of molecular biology, which consists of the translation, transcription, and translation, transcription. Okay, and DNA replication also. After these things, we have discussed about the molecular biology. We have discussed about various coenzymes, cofactors, what are their roles, what are their importance, various examples. Followed by, we have moved toward the more into the medicinal chemistry, the drug discovery and development process. What are the stages we have discussed? We have discussed about various neurotransmitters. We have discussed about the hypertensive medications. What are the classifications and how the design of the hyper inhibitor of angiotensin. We have all discussed it. Then we have discussed about the antibacterial agents. Majorly, we have discussed about penicillin and other beta-lactamase antibiotics. Also, we have discussed about various classes of antibacterial agents. Then we move toward the polyketide biosynthesis. We have discussed various topics related to the antiviral drugs. We have discussed about the virus life cycle. Then what are the different targets which can be, which can be useful for our this antiviral drug discovery. And then we in the last class, which was yesterday, we have discussed about the cancer chemotherapy. We have discussed about the various targets which are can be used to have a anti-cancer drug like intercalating agents, chain cutter. Then we have discussed about the alkylating agents, topoisomerase inhibitors, and so on. Then we've discussed about cholesterol biosynthesis, the QSAR, little bit about. So these are the things which we have learned so far in our course, and most of the topics somewhere so there were some topics which I could not discuss in much detail. So today in today's class I would like to do those things. Okay, if you have any doubt or any query regarding any of these classes, you can let me know via email also or via the discussion forum. Okay, here is, if you just go to this place, if on the question answer session, you will find there is a discussion forum. Here you can ask any problem you have regarding any, I mean, live sessions or any general topics from the course. You can also discuss. We can also discuss. Okay, I will share my email ID at the end. So after today, we will communicate. We can communicate through the email. If you have any doubt or any query, I will try my best to answer your queries. Okay. So very good afternoon to all. To the last class, to the summary session for the NPTEL course of Organic Chemistry in Biology and Drug Development. My name is Mousumi Deb and I'm a PMRF Fellow from the Department of Chemistry and Chemical Biology, IIT ISM Dhanbad. Today it is our summary session. Okay. So today what I will do is I will summarize few topics from your course. These topics I think I have not I have discussed this in the class, but a few topics are we have discussed in very beginning. So I just want to give you a recap of all these things, and a few topics some students have mailed me that they want to some clarification or about few topics, so that also I will discuss. And there are few things which I could not discuss in our course because this was not very much in detail in your syllabus, but these are very important in the aspects of the organic chemistry's application in the biological sciences. Okay. So let me start the session. After in between, I mean, so today there is no question answer like we everyday do. So I do not have any question today, but I have a few things which I will discuss, and in between I will check the chat box. So if you have any doubt or any query, please let me know when I will check the chat box. Okay. In between, I will keep checking. So if you have any doubt or query, please let me know. So let us start. So basically, we have started our course with the amino acids, which are the basic of our life. So there are crucial amino acids. There are essential, which are known as the essential amino acid, and 9 amino acids which are essential for human, because we human cannot synthesize this amino acid in our body. We have to rely upon other sources like the food sources, and we have to take this amino acids out from outside, because our body cannot synthesize it. So what are these amino acids? These amino acids are the phenylalanine, which is crucial for the neurotransmitter production, impacting brain function, mood regulation. We have valine, crucial for muscle repair, growth, energy, and blood sugar and immune system. Then we have threonine, crucial for the body function, nervous system, health system, collagen and tissue formation and maintenance. Tryptophan is crucial for serotonin production, mood regulation, and so on. Methionine is crucial for methylation as a precursor of antioxidant and detoxification. Then we have leucine, which is crucial for the muscle growth, repair, and regulatory blood sugar and providing exercise energy. Isoleucine, crucial for muscle repair, endurance, and regulating blood sugar during exercise. Lysine, crucial for growth, tissue repair, healthy immune system. And histidine is important as it is a it is a precursor of key compounds of the physiology. Classification of amino acids. So amino acids can be classified via various technique. I mean, there are various parameters by using amino acids can be classified. There is a classification of amino acid based on the polarity of the side chain is one of those. So the amino acid, if you do this by using the polarity of the side chain, so there are two type of amino acid, which is polar and then non-polar one. Polar one, where the side chain is polar, which contains a OH group like serine, threonine, tyrosine, SH group like cysteine, amide group in glutamine, asparagine, NH2 group in nitrogen as a basic amino acid in lysine, arginine, and histidine, and carboxylic group like acidic amino acid in aspartic and glutamic acid. So all these amino acids have the polar side chain. So they are part of a polar amino acids where this hydrophilic polar side chain are can form the hydrogen bond with water. Okay, and there are some non-polar amino acids. We do not have any kind of hydrophobic group, the hydrophilic group, but these hydrophobic side chains are present. 9 amino acids are non-polar like glycine, alanine, valine, leucine, isoleucine, phenylalanine, tryptophan, proline, and methionine. Okay. Then we have discussed, so these amino acids, they join together to form a peptide chain, which is a primary structure of a protein, followed by the secondary structure of a protein, which are the alpha helix and beta pleated sheet, and we also have beta bands. Okay, so that is when the all the polypeptide chains, they interact with each other with the help of the hydrogen bonding. They form the secondary structure of the protein, which is the basically the alpha helix and the beta pleated sheet, right? And then these alpha helix and beta sheets, they can interact with each other via various interactions like van der Waals interactions. In case of secondary structure of the protein, primary interaction is the hydrogen bonding interaction. But as we move toward the secondary and tertiary structure of the protein, there are several other interactions like van der Waals interaction, pi stacking interaction. We have the hydrophobic interactions and salt bridge interaction, disulfide bonds, and so on. So these form the tertiary structure of the protein. So the tertiary structure is basically the either the fibrous one and the globular one. So this fibrous one is a class of insoluble protein which constitute main structural elements of our body. It has a long and narrow shape, have a helical or sheet structure. It basically forms the secondary structure, consists of a repeating amino acid sequence. It is insoluble in water, acid, and bases. Have strong intermolecular interaction, less sensitive to changes to temperature and pH, involved in forming structures like collagen, elastin, actin, myosin, fibrin, and keratin. And globular proteins are the protein that are soluble in water, tend to be involved in metabolic functions, has a round and spherical shape, have a folded ball-like structure, and basically the tertiary structure are the things which form this globular protein. They consist of an irregular amino acid sequence, soluble in water, acid, and bases, have weaker intermolecular interaction, which are sensitive to the changes of the temperature and pH, involved in the metabolic reactions. Enzyme, hormone, hemoglobin, immunoglobulins are the example of the globular protein. So fibrous and globular protein, we actually see in that our hair is made of collagen, our skin is made of elastin. So all these are fibrous proteins. So these are not soluble in water, right? Our hair or the skin, this doesn't get dissolved when we add them in the water. But globular protein, the main importance of the globular protein is that they are water soluble and they consist of the various important enzymes, hormones, and of our body. Okay. Then the protein, it has a zwitterionic, the amino acids, they have a zwitterionic structure. We all know it has a amine and carboxylic acid group in its structure, and the amine actually it stays as a amine ammonium ion NH3 plus, and the carboxylic acid it basically stays as a COOH minus, so which is known as the zwitterion, which is like this. Okay, so this is the zwitterionic structure of the amino acid. And the zwitterionic structures, so this particular zwitterionic structures or the ionic form of the amino acid can be seen while the changing the pH. Okay, so that is also I have discussed earlier that how the charges are changed. Okay, so net charge is 0 when there is a positive negative charge, net charge of 0. So this is known as the isoelectric point, right? This is known as the isoelectric point of the amino acid when it does not move at any side of the electron, not the positive, not the negative end. Okay, so isoelectric point is the pi, should be average of the pK values of two most acidic amino group. So sometimes what happens, so this is one example. So this is, as you can see, is an acidic amino acid. So whenever the amino acid is the neutral one, it has only a ammonium group, NH2 group, and a COOH group, and both has its particular pKa values, right? So pi value of these amino acids will be at the average of pK1 and pK2, right? But when there is a acidic amino acid or basic amino acid, the thing changes, right? So as you can see, in case of the acidic amino acid, we have two carboxylic acid group, and the net charge is 0 between pK1 and pK2, which are these two, which are the lower pK values. So in case of acidic amino acid, to have isoelectric point, you have to average the lower two pK values, and in case of the basic one, you have to average the higher pK value. All these things I have discussed in our course in detail. You have to just go to your first week slides or the second week slide. You will find there are detailed example with the acidic amino acid, neutral and basic amino acids, how to calculate their isoelectric point. So this is a very important topic in your syllabus. So this is the example. This is the example with the acidic one. As you can see, the net charge is zero. In between the positive and negative, two lower pK values. Okay, and in case of the basic one, the higher pK values you have to average to have this particular pi value. So in case of the acidic amino acid, pK1 and pK2 are the which you have to average. In case of the basic amino acid, pK2 and pK3, that you have to average to have the isoelectric point. Okay, all these I have discussed in detail in your slides. As I am saying, you have to just go on. So is there any doubt or anyone have any problem while solving the how to solve the isoelectric point or something? Okay. So if you have any doubt how to solve the isoelectric point, you can let me know. Okay. Although I have shown it in detail, so you have to just go to your previous slides and you have to practice. Okay. So now let me discuss a few little bit about the enzyme activity and enzyme. So enzyme kinetics I have discussed in detail. These topics are very important, like the Michaelis-Menten kinetics, enzyme kinetics, and the Michaelis-Menten equation, the mathematical questions that we have solved during the classes. Okay, so we have the Lineweaver-Burke plot and other. There are several plots like the Hans-Wolff plot. We have Lineweaver-Burke plot. Okay, all these plot that maybe I mean mathematical questions can come from that all these topics. Okay, so now when we were discussing about the enzyme kinetics, one thing which we have discussed was the enzyme inhibition. Okay, how the enzyme inhibition happens. So enzyme are the natural catalyst, right? Our body, in our body, the biological catalyst are the enzyme which actually fasten up the various biological processes. Enzymes are basically the proteins, and they have all the characteristics of the protein, and and the enzymes they actually follow a particular kinetics, which is the Michaelis-Menten kinetics, where first when the amount of the substrate is lower, the enzyme the rate of the reaction is with first order with our substrate concentration, then it goes to the mix, and then it goes to the zero order. I hope you remember how the curve of the Michaelis-Menten looks. Okay, and this in there are two theories. We all know that the one is the lock and key theory, and there is a induced hypothesis theory. Lock and key theory is the most basic one, which says that the shape size or the shape of the particular active site of an enzyme is very similar, exactly same with the size of the substrate, and the substrate and the enzyme, the active site, they fit each other with each other in a very perfectly like a lock and key. Okay, but in the induced fit hypothesis, it is the most acceptable one actually, which says that the active site is not exactly as the same shape of the substrate, but when the substrate is nearby the enzyme's active site, they actually change itself a little bit to accommodate that particular substrate, which is this. So this induced phase hypothesis is the most acceptable one. Okay, and enzyme kinetics is very important, the enzyme turnover number and all those things which we have discussed. So there is a thing which is enzyme inhibition. So many enzymes, so there are two type of inhibition, reversible and irreversible. So in the reversible inhibition, we have the many inhibitor reversibly binds to the enzyme molecule, affecting the equilibrium constant of the reaction. Several types of such inhibitions are known and can be distinguished with the help of the Lineweaver-Burke plot of the enzyme kinetics into three subtypes. They are competitive inhibition. The competitive inhibitor is structurally related to the substrate, and it competes with the substrate for the active site of the enzyme. It binds reversibly at the active site of the enzyme to form an enzyme inhibitor complex. Once the inhibitor occupies the active site of the enzyme, it prevents binding the substrate to form the enzyme substrate complex. Thus abolishes the formation of the normal and metabolic product. Inhibitor binds reversibly the enzyme because the competition can be decreased simply by adding more substrate. The equation for enzyme catalyzed reaction in presence of the competitive inhibitor as follows this. Kinetically, the inhibitor binds with the free enzyme reversibly to form enzyme inhibitor complex, which is catalytically inactive and cannot bind with the substrate. The competitive inhibitor reduces the availability of the free enzyme for the substrate binding. The total enzyme exists in the reaction medium as the free enzyme and the bound enzyme in presence of the inhibitor, and Michaelis-Menten constant of the normal reaction is to increase the nu Km, and then the reducing constant in this. The Michaelis-Menten equation for the competitive inhibition becomes this one. Okay, consequently the rubber reciprocal form or the Lineweaver-Burke equation is this one. In presence of the competitive inhibitor, the slope of the double reciprocal plot is increased, and by the intercept value is the remains this. Okay, so these are the values of the slope and intercept. So these values you do not need to memorize. Okay, this can be derived from this equation itself if you just know the steady state. But these things, the complete I mean deduction, how it happens, it is not in your syllabus directly. But if possible, you can remember the slopes, the values of the slope and the intercept, so that if there is any kind of question is given, you can solve it easily. Then we have non-competitive inhibition. Non-competitive inhibitor does not have any structural similarity to the substrate, but it binds both the free enzyme and enzyme substrate complex. The simplified equation of steady state kinetics is goes like this. Okay, as you can see, enzyme and inhibitor forms enzyme inhibitor complex, which in presence of substrate from the enzyme substrate inhibitor complex. Enzyme and substrate from the enzyme substrate complex, which in presence of inhibitor again form enzyme substrate inhibitor complex. And the enzyme substrate complex we know gives us the free enzyme and the product. The presence of the substrate has no influence on the ability of non-competitive inhibitor to bind the enzyme and vice versa. Its binding alters the conformation of the enzyme and reduces its catalytic activity due to changes in the nature of the catalytic groups of the active site. EI and ESI complexes are non-productive. On increasing the substrate of the saturated concentration does not reverse the inhibition, leading to unaltered Km but a reduced Vmax. Reversible of the enzyme requires special treatment like dialysis or pH adjustment. This is very important, that the reversal of the inhibition of in case of a non-competitive inhibitor, it required a special treatment like dialysis and pH arrangement. But in case of our competitive inhibitor, so what happens in case of competitive inhibitor, it has the the inhibitor molecule looks or the size and shape is similar to a substrate. So there is a competition like for this in between the substrate and inhibitor to bind with the active site of the enzyme, where when most of the inhibitors they bind to the active site of the enzyme, let's say they win the competition and bind to the active site of the enzyme. So to reverse the effect of this inhibitor is very simple. You just have to increase the amount of substrate. If the amount of substrate is increased, there are more chance of with the substrate molecule to bind into the active site rather than the inhibitor molecule. So the reversal of the competitive inhibitor is only to increase the substrate concentration. But in case of the non-competitive inhibition, it is not so. Okay, on increasing the substrate to a saturating concentration does not reverse the inhibition. Does not, considering the steady state kinetics of the non-competitive inhibition, there is a dissociation constant or inhibitor constant. Okay, which is this one. This is very important, that this is a dissociation or inhibitor constant, which looks like this. So now the total enzyme concentration is E0 equal to E plus EI plus ES plus ESI and so on. So now the equation 2 can be equation 2 which is in the last here. Okay, so equation 2 now can be rewritten as this one, and it can be expressed like this, and this is the value of the this is the value of the Michaelis-Menten equation in case of the non-competitive inhibitor, and it is the double reciprocal follow. Again, I am saying the derivations are not in your syllabus, so I am not discussing the derivations in detail, but just if possible, there was a chart which I don't know, I don't exactly remember in which week I have discussed this. I think the enzyme inhibition we have discussed in our some in between like third or fourth class we have discussed. Then there I have mentioned a chart which contain the altered values of the Km and Vmax in case of various enzyme inhibition. That chart, if you possible, you can remember. Okay, then we have a uncompetitive inhibition. Uncompetitive inhibitor has no structural similarity to the substrate. It may bind to the free enzyme or enzyme substrate complex that exposes the inhibitor binding site. It binding although away from the active site causes structural distortion of the active or allosteric side of the complex enzyme that inactivates the catalysis. This leads to decrease in both Km and Vmax. This inhibition cannot be reversed by increasing the substrate toward the saturating concentration, and the reversal requires special treatment like dialysis. This type of inhibition is rare and is countered as the multi-substrate enzymes, where the inhibitor competes with one substrate to which it has some structural similarity is uncompetitive. So the reaction goes like this, and the steady state kinetics looks like this. Okay, the E plus S forms ES, which is with inhibitor it forms the ESI, and without inhibitor it forms it gives us the enzyme and the product. Considering the steady state kinetics of the uncompetitive inhibitor, the dissociation constant Ki is this, and here this is the Michaelis-Menten equation, and the double reciprocal value goes like this, and this is the double reciprocal. This is the plot. Okay, so the plots are very important in case of our I mean the competitive, non-competitive, uncompetitive. I have shown you how this double reciprocal plot or Lineweaver bark plot looks like, right? So questions can come from these graphs also. And I think there was a question which I have discussed in a week, where I have discussed that how this Km value and Vmax value changes in case of this inhibitions. Okay, so this you have to remember. So this one is the for the without inhibition, it is with inhibition. Here also this is without inhibition, it is with inhibition. So these plots you have to remember. So now we will move toward the next problem. If you have any doubt or any query, you can let me know. Okay, you can let me know in between, or at last you can let me know. Now let us go to the next slide. So let us see what are the examples. So some examples of the competitive inhibitors are the sulfonamide, a simplest form of the sulfa drug. It is an antibiotic useful in treatment of some kidney infection. As a structural analog of para-aminobenzoic acid or PABA, the sulfonamide binds, inhibits the bacterial growth. PABA is a structural part of folic acid. The sulfa drug is highly toxic to bacteria and must synthesize their own folic acid. Since humans require folate from dietary source, the sulfonamide is not harmful at the dose that kills bacteria. Methotrexate is another competitive inhibitor for dihydrofolate reductase. The drug closely resembles to the folic acid and is used as an anti-cancer anti-metabolic chemotherapy, particularly for pediatric leukemia. It hinders the availability of the tetrahydrofolate, which is important for anabolic pathways, particularly synthesis of purine nucleotides for the DNA replication. I think I have discussed when I was discussing about were discussing about this antibacterial agents. We have discussed about the I mean sulfonamides and in detail. Okay, and also I have mentioned there that sulfonamide are the very good example of our what should I say, competitive inhibitors. Okay, so in that particular classes, I think it is our week 10 course content. You will find it there where I have discussed about the sulfonamides as the competitive inhibitor for this particular enzyme. Okay. Examples of the non-competitive inhibitors are mostly the poisons. Okay, cyanide and the azide inhibit the enzymes with the iron and copper as a component of their active site or the prosthetic group. For example, cytochrome C oxidase. Cyanides bind with the Fe3 plus in the heme of the cytochrome A3 component of cytochrome 3 oxidase, prevents the electron transport to oxygen. Mitochondrial respiration and energy production cease and the cell death rapidly occurs. The central nervous system in the primary target of the cyanide toxicity. Acute inhalation of the high concentration of the cyanide during the smoke inhalation during the fire or automobile exhaust provokes the brief central nervous system stimulation rapidly following the convulsion, coma and death. Acute exposure to the lower amounts can cause lightheadedness, breathlessness, dizziness, numbness and headaches. Cyanide is present in the air as hydrogen cyanide, in soil water as the cyanide salts, and in foods as cyanoglycosides. Uncompetitive reversible inhibition is rare but may occur in multimeric enzymes. Some examples of uncompetitive reversal inhibitors are the inhibition of the K regulatory heme synthetic enzyme, the delta amino valvinate synthase and dihydrate, and the heme synthase of the very by heavy metal ion like lead. Heavy metals like lead form mercaptides with SH in the active site of the enzyme. The oxidizing agents like ferricyanide also oxidizes SH to disulfide linkage. Ability of the lithium to exert the profound and selective psychopharmacological effects has been focus of the considerable research effort. There is a increasing evidence that lithium exerts its therapeutic action by interfering with the phosphoinositide metabolism in the brain and prevention of the inositol recycling by an uncompetitive inhibition of inositol monophosphate. Few pesticides are uncompetitive inhibitor. The best known example being the herbicide N-phosphomethylglycine, commonly known as the glyphosate or Roundup, an uncompetitive inhibitor of 3-phosphoximate 1-carboxyphenyl transferase. Okay, so these are the various examples. Then we have another thing which is the irreversible inhibition. Irreversible inhibitors have no structural relationship with the substrate and they bind covalently with the active site of the enzyme and destroy the essential functional groups of the active site. So irreversible inhibitors are used to identify functional groups of the enzyme active sites at the location they are found. Subsets of the irreversible inhibitors called the suicide irreversible inhibitors that are relatively inactive compounds. They subsets they get activated upon binding with the active site of the specific enzyme. However, it does not release any product because of its irreversible binding at the active site. An enzyme in effect commits suicide by activating the inhibitor. Due to its very nature, in suicide irreversible inhibitors are also called the mechanism based activator or transition state analog of the inhibitor. So this is important. Okay, I think I have told you earlier also that the suicide inhibitors or irreversible inhibition is known as the mechanism based inhibition and transition state analogs of the inhibitors. Okay, the transition state analogs are extremely potent and specific inhibitors because they have high affinity and stronger binding to the active site of the drug target enzyme and natural substrate product. However, exact design of the drugs is precisely mimic the transition state is a challenge because unstable structure of the transition state of the enzyme catalyzed reaction. Kinetically, the irreversible inhibitor decrease the concentration of the active enzyme and in turn decrease the maximum possible concentration of the enzyme substrate complex. It reduces the initial concentration of E0 until all enzymes are consumed to E0 minus E0 level. Once the process between enzyme and inhibitor goes completed, there is no free inhibitor is present in the mixture. Therefore, further addition of the substrate follows the Michaelis-Menten equation. In absence of inhibitor, the rate of the product formation with the excess substrate is this Vmax equal to Kcat into E0, while in presence of inhibitor, the formation of the excess substrate is Vi max equal to Kcat E0 minus I0. Here Kcat is the turnover number which is defined as the maximum number of molecule that an enzyme can convert to product per catalytic cycle. This is important. What is the turnover number? I have discussed earlier. I have also shown you mathematical question related to the turnover number. So this is also very important. Mathematical questions can be asked related to the turnover number also. Combining the two equations gives us this. This is the about this equation is about the irreversible inhibitor. Some clinical examples of suicide inhibitor is NN-dimethyl propargylamine. This compound is an inhibitor of the enzyme monoamine oxidase. Monoamine oxidase is responsible for breaking down neurotransmitters such as the dopamine, serotonin, thus decreasing the concentration in the brain. Diseases such as the Parkinson disease, depression occur because of the decreased level of dopamine and serotonin. So there is another thing which I could not discussed earlier in our class, but it is very important, which is the cholinergic signaling system. Okay, so what is the cholinergic? So we have learned about the we have heard about the acetylcholine a lot. Okay, and so what happens? So first look at what happens in the synapses. Okay, involving acetylcholine as a neurotransmitter. So acetylcholine is a very important neurotransmitter. So here you can see there is a acetylcholine. Okay, and there is a this is the choline, this is the acetic acid which forms the acetylcholine, which is this one. Okay, then this acetylcholine goes into the vesicle and after the at the end of the synapse. So what happens that this acetylcholine is released and it is taken into the prosynaptic nerve. So it shows that the synapse between two neurons and the events that involved when a message is transmitted from one neuron to another. The same general process takes place when a message is passed from neuron to muscle cell. The first stage involves the biosynthesis of acetylcholine. Acetylcholine is synthesized from the choline and acetyl coenzyme A. This is the reaction. Okay. Choline and acetyl coenzyme A, which with the help of the enzyme choline acyl transferase from the acetylcholine, this one. Acetylcholine is incorporated into the membrane bound vesicle by the means of the specific transport protein. The arrival of the nerve signal leads to the opening of the calcium ion channels and an increase in the intracellular calcium concentration that induces the vesicles to fuse with the cell membrane and release the transmitter into the synaptic gap. The acetylcholine crosses the synaptic gap, binds to the cholinergic receptor, resulting in the stimulation of the second neuron. Acetylcholine moves to an enzyme called the acetylcholine esterase, which is situated on the prosynaptic neuron. It catalyzes the hydrolysis of acetylcholine to produce choline and acetic acid. Choline is taken up by the presynaptic neuron by the transport system to continue the cycle. So as you can see here, it is recycled into the choline and the choline is again taken up to continue this particular cycle. So this is the cholinergic system and how the acetylcholine acts as a neurotransmitter and what happens. So the cholinergic nervous system involve nerves which use the neurotransmitter acetylcholine as a chemical messenger. These include the motor nerves which innervate skeletal muscle, nerves which synapse of the other nerves in the peripheral nervous system, the parasympathetic nerves innervating cardiac and smooth muscle. There are two types of cholinergic receptor: muscarinic receptor, which is present in the smooth and cardiac muscle, and nicotinic receptor present in the skeletal muscle and the synapses between the neuron. Acetylcholine is hydrolyzed by the enzyme acetylcholine esterase. When it departs the cholinergic receptor, the hydrolytic product choline is taken up into the presynaptic neuron and acetylated back to the acetylcholine. The cholinergic receptor and the enzyme acetylcholine esterase are useful drug targets. Acetylcholine cannot be used as a drug because it is rapidly hydrolyzed by the acid and enzymes. It shows no selectivity for different types and subtypes of the cholinergic receptors. Then this is the cholinergic receptor. There are two types. One is ligand-gated ion channel and G protein coupled receptors. The ligand-gated ion channels are like nicotinic acetylcholine receptors, and the muscarinic receptors are of five types: M1, M2, M3, M4, and M5. Whereas the nicotinic one is the neuronal nicotinic acetylcholine receptor and the skeletal or muscular. Okay, and the neuronal ones present in central nervous system, autonomic ganglia in the peripheral nervous system, whereas the muscle one present in the skeletal muscle of the neurotransmitter junction. Neuronal one are of two type, homopentameric and heteropentameric. Okay, and it combines of the several subunits. The neuronic subunits goes to the alpha and beta subunits. So these are the different cholinergic receptors. So till now, if you have any doubt or any query, let me know. Now we will move toward different topics. Okay. So these were few topics of the, there are more in biological aspects which we had not discussed in our course in detail, some topics. Okay, most of the topics I have already discussed and all are given in your particular slides. I hope you know where to find the slides. Okay, so I will just show you at the last where to find all the materials. I hope, I think you know, but still I would try to give you an overview about where to find all the things. Okay, now let me go to the next slide. So there are very important thing which we had not discussed in details is the pharmacokinetics and the pharmacodynamics. So when we were discussing about the various biological system, we were discussing about the drug discovery process. So two things are very important whenever you are dealing with the organic chemistry, with biology and the biological sciences. So one is the pharmacodynamics, another is the pharmacokinetics. Okay, so pharmacokinetics is the basically pharmacodynamics is the study of how drugs interact with the molecular target, means the effect of the drug on the body, whereas the pharmacokinetics is the study how the drug reaches to the target, various and what is the particular journey of a drug in our body, like the absorption, distribution, metabolism and excretion. Okay, so pharmacokinetics is basically how our body is what our body is doing to a drug. So the first thing, so this is there is a very famous term which is known as ADME, ADME. Okay, ADME is the term which is widely used to discuss to indicate the pharmacokinetics. What is the ADME? ADME. So ADME or admit. Okay, so admit the what is the full form of it? A for absorption, D for distribution, M for metabolism, E for excretion, and T for the toxic effect. So this is the admit is widely used to discuss the pharmacokinetics. So first is the absorption and the distribution of the drug. So first is the drug administration. So a drug can be given to you via oral via tablet or capsule or any syrup or drops. Then by the intravenous by using the injection. It can be given by intraperitoneals or subcutaneous, intramuscular or inhalation. Okay, so these are the various modes of the drug administration. After a drug is administered, it is absorbed via membranes of our oral cavity and gastrointestinal tract and peritoneum, skin, muscles and lungs. Then after absorption and distribution, the drug goes to the plasma with the for plasma protein binding. Then the drug goes to the liver for then after binding with the target site, the neuron receptors, bone fat for the inactive storage deposits, and after it goes to the liver for the metabolism. The metabolites, they are excreted by intestine, kidneys, lungs, sweat glands and etc. Okay, from liver also it can be excreted directly. And so this is the administration, distribution, metabolism, excretion. This is the ADME, and ADME T is the toxic effect. So sometimes that particular drug or a particular agent can have some toxic effect on our body. So all the studies which are related to the pharmacokinetics are known as the ADME study or ADME study. Now, what is pharmacodynamics? Pharmacodynamics is the basically how the drug is acting in our body. So there are various receptors which are responsible for the pharmacodynamic effect. There are protein molecules with one or more binding site located on the cell membrane. They receive a signal from the body's chemical like neurotransmitter, hormone, and enzymes. Signal will cause a molecular event for inside the cell to occur. Drugs may enhance, diminish, or block the generation like agonist, partial agonist, antagonist. Affinity is the how we attract the attraction between drug and receptor. High affinity drug will bind easily with the receptor. Low affinity requires higher concentration of the drug to get a therapeutic response. Drug potency, amount of the drug required to produce therapeutic response. The dose response curve ED is the effective dose amount of drug that produce a therapeutic response of 50% of the people taking it. Toxic dose amount of drug that produces adverse effect by 50% of the people taking it. Therapeutic index or TI is the margin of safety ratio between TD and ED, and high the therapeutic index, safer is the drug considered to be. In general, non-prescription drugs have much high TI than prescription drug. So these are the pharmacokinetic parameters. Like peak concentration is the peak plasma concentration of the drug after administration. The amount of the drug when it reaches to the maximum concentration in the plasma. It is also known as the maximum plasma concentration. Peak time, the time when the drug concentration reaches to the maximum after the drug administration. At this point, the rate of the drug absorption is equal to the drug elimination. Now, then we have the terminal half-life, the time required for the concentration of the drug to reach the half of its original value. Area under the curve, the integral of the concentration time curve after the single dose of the safety of a steady state. It is a measurement of how extend the drug bioavailability. It shows the total amount of the active drug is bioavailable for the systematic circulation, and clearance is the rate at which the drug is removed from the body of drug per unit time. So is it clear? Before moving, further, I want to know that is it clear or is there any doubt? The pharmacokinetics and pharmacodynamics, if there is any doubt, please let me know. Otherwise, I will move forward because now we will enter to summarize the various therapeutic effects, okay, or various therapeutic agents. Okay. If there is no doubt, then I am moving forward. Please let me know. So, since today it is our last class, so if you have any doubt or any query, please you can let me know. At the end also you can let me know. Then I will just discuss that particular topic at the end. Okay. Till now, whatever I have discussed, do I need to repeat anything? Okay. So if there is no doubt, then I am moving. So one more thing which I am going to discuss today, I think I have discussed it a little bit, but not in detail, is the different regulatory bodies. So whenever there is a drug which is in market, then before going to the market there are different governing bodies by which the drug has to pass before going to the market. So the most important one is the FDA, which is the Food and Drug Administration of the USA. Okay, so this is I have discussed when I was discussing the drug discovery stages. So there was a point when the drug goes for the FDA approval. So FDA is the federal agency of the United States of the Department of the Health and Human Services, one of the United States federal executive departments. The FDA is responsible for protecting and promoting public health through the control and supervision of the food safety, tobacco product, dietary supplements, prescription and over-the-counter pharmaceutical drugs and medication, vaccine, biopharmaceutical, blood transfusion, medical devices, electromagnetic radiation emitting devices, and so on. Similar to FDA in India, we have several governing bodies like Central Drug Standards Control Organization or CDSCO under the Director General of the Health Services, Ministry of Health and Family Welfare, Government of India, and FSSI, which is the Food Safety and Standard Authority in India, which is also governed by Ministry of Health and Welfare of Government of India. In Europe, we have this European Medicinal Medicine Agency or EMA, and MHRA is present Also, in case of the Kingdom, the medicines and healthcare products of the regulatory agency, executive agency of the United Kingdom is responsible for ensuring the medicine and medical devices work are acceptably safe. In Japan, we have this PMDA, which is the Pharmaceuticals and Medical Devices Agency, and is a Japanese government organization similar to the US FDA and or US FDA or Indian CDSCO. So this is the different. So these are the different governing body. Before a drug goes to the market, they have to pass through these governing bodies, various. Okay, and then they must go to the market. Okay, so pharmacodynamics. Okay, sure, I will discuss it. So pharmacodynamics, as I was saying, whenever we are taking a drug in our, so there are two things. One, what our body is doing to a drug, and the second is what this particular drug is doing to our body. So what our body is doing to a drug is known as pharmacokinetics, and what the drug is doing to our body is known as the pharmacodynamics. As I have mentioned, that the taking of the drug, the administration, distribution, all these things are pharmacokinetics, and the pharmacodynamics is the effect of the drug. Okay, how the drug behaving in our body, how it is binding to the active site, how it is binding to various receptors of our body, like various neurotransmitter, hormone, enzyme, how it is signaling the various things. So this is known as this study is known as the pharmacodynamic study. Okay, the drug, it may enhance the effect of a particular generation of a signal of a particular activity of a receptors. Those drugs are known as the agonist. The drug can diminish the effect of a particular receptors. Those drugs are known as partial agonist. Some compound or drugs, they can completely inhibit a particular neurotransmission or some generation of a signal from a receptor. So those are known as the antagonists. So there are several terms which are very important whenever there is a pharmacodynamic study. Okay, so what are the terms? So there is the terms are the affinity, which is the attraction between the drug and receptors. How they are, how much is it binding easily or is it not binding easily? If the particular drug is binding very easily, so it is known as the high affinity drug, means lower concentration of drug is fine. But if a drug has a lower affinity, means more concentration of the drug is required, okay, to have the therapeutic index. So these are the various terms which are important. Then we have the drug response curve, and I don't know if you remember, I have when I was discussing about the therapeutic index in our I think it was in our ninth class, we were discussing about the therapeutic index, which is the various. So there is various terms which are very important in the pharmacodynamics. In a broader aspect, thermodynamics is what the particular drug is doing to our body. What is the effect of the drug on the various receptors or enzymes or various things. So there are several parameters by which this pharmacodynamic effects can be measured. One is like effective dose. Okay, then is a toxic dose. How much drug is required for 50 generating a toxicity effect or effective effect. Okay, and then we have this IC50 value, EC50 value, all these things are present there. Like for suppose a drug is an enzyme inhibitor, then IC50 value is measured to have to know the pharmacodynamic effect of the drug. What is the IC50? Is the concentration is how much of the drug is required to diminish the enzyme's activity to 50%. So these are various parameters which measure the pharmacodynamic effect on our body. So this is the pharmacodynamics. All these things, all this this terms and the this agonist, antagonist, I think I have discussed, but I have not mentioned earlier in our previous classes that all these things are falls under the pharmacodynamics. So this is about the pharmacodynamics. So is it clear what is pharmacodynamics, or do I need to repeat one more time? I hope it is clear now. Please let me know, otherwise I will discuss it more. Anyway, I will then if there is no doubt regarding this, I will move forward. Okay. So as I was saying that these are the various governing body, and the drugs must pass through these governing bodies before going to the market. Okay. And US FDA is one of those, and there are several other governing bodies also. So this I have discussed earlier. So now I will just give you a quick recap to the various therapeutic effects drugs which we have discussed earlier. So we have discussed about the antibiotics, right? So there are various classes, like penicillin is the most important one. So, so there are different several generations of the penicillins, like penicillin G, amoxicillin, ticarcillin, piperacillin. So this is the main structure of penicillin. The remembering the structure of penicillin is important, but there may be question come from the structure itself with the penicillin. Okay, so only the basic structure of penicillin, cephalosporin, quinolones are important. Tetracycline and macrolide, or their structures are little difficult, so you do not need to memorize, but you should know how it looks. Okay, now for suppose the question is given to you by only giving you a structure, you at least need to identify that it is a structure of a macrolide or tetracycline. That's all. Okay. So penicillin, the several generations of penicillin are like penicillin G, we have amoxicillin, ticarcillin, piperacillin. Cephalosporin like cephalotin, ceruoxime, ceftazidime, cefepime. Quinolones, we have nalidixic acid, ceprofoxacin, levofloxacin, moxifloxacin. Macrolides like erythromycin, clarithromycin, telithromycin. Tetracycline, we have oxytetracycline, doxycycline, and ticarcycline. So various mechanism of action of antibiotics is very important. So this figure, I like this figure very much because it summarizes complete thing. So there are in antibiotics which affect the cell wall synthesis of the bacteria. So what are these? These are the cycloserine, vancomycin, bactricin, penicillin, cephalosporin, monobactam, carbapem. There are some which inhibit this folic acid metabolism, which is very crucial, like trimethoprim and sulfonamide. Sulfonamide inhibit the para-aminobenzoic acid, and it hindered the conversion of para-aminobenzoic acid to DHF, and DHF to THF conversion is inhibited by trimethoprim. There are cytoplasmic membrane structure and function. The inhibitors are the polymyxin and actinomycin. So these are the lipid biosynthesis is inhibited by the plantanomycin. Then protein synthesis, the tRNA is inhibited by the mucopurocin and puromycin. Protein synthesis, the 30S inhibitor. So we all know the ribosome, bacterial ribosome contain two subunits, one is 50S and one is 30S. The 30S subunit inhibitors are like tetracycline, spectinomycin, streptomycin, gentamicin, kanamycin, amikacin, and nitrofurans. And 50S inhibitors are the erythromycin, chloramphenicol, clindamycin, then lincomycin. DNA-directed RNA polymerase inhibitors are the rifampicin, streptofadicins. RNA elongation are done by the actinomycin. DNA guidage is inhibited by the nalidixic acid, ciprofloxacin, and novofoxacin, which are the part of the quinolone class of the compounds. Okay, so these are the various I mean, this figure actually summarizes various mechanisms of inhibition for the by which an antibacterial agent can exert its antibacterial activity, along with the examples. Okay, so remembering this thing is very important, all the mechanism of action along with their examples. Then let us discuss about a little bit about the antifungal drugs. Although it is not very much in detail in your syllabus, but antifungal drugs are very one of the very important therapeutic agents which we use. Okay, so there are several classes of the antifungal drugs. So like imidazole, triazoles, imidazole containing antibacterial antifungal drugs are like cotrimazole, oxyconazole, miconazole, iconazole, ticonazole, and ketoconazole. Now, triazole containing are the fluconazole, itraconazole, tarconazole, voriconazole, isavuconazole, and posaconazole. So all these azole containing antifungals, they inhibit the lanosterol 14 alpha demethylase, the enzyme required to convert the lanosterol to ergosterol. Okay. Then we have the griseofulvin, which inhibits the mitosis in the dermophytes. This is the ineffective when applied topically. We have the echinocandis or CAM, which is the caspofungin, endulafungin, and micofungin, which inhibit the cell wall synthesis by targeting glucans. Okay, then we have the flucytosine, the pyrimidine analog converted into the 5-fluorouracil by fungal enzyme cytosine deaminase. They active against the yeast infection. We have the pollines like natamycin, amphotericin, nestin, and aligned amine, which are the amphorin, and this is inhibit the squalene epoxidase. So mycosis is the fungal infection in the animals and humans. Mycosis are the common and variety of environmental and physiological condition can contribute to development of fungal diseases. The triazoles are very important actually as an antifungal agent. So as you can see, fluconazole, albuconazole, voriconazole, like other imidazole and triazole class of antifungals, fluconazole inhibits the fungal cytochrome P450 enzyme. 14 alpha demethylase, mammalian demethylase activity is much less sensitive to the fluconazole than the fungal demethylase. This inhibition prevents the conversion of the lanosterol to ergosterol, an essential component of the fungal cytoplasmic membrane, and the subsequent accumulation of 14 alpha methylsterol. Fluconazole is an inhibitor of the human cytochrome P450, particularly the isozyme CYP2C19. In theory, therefore, fluconazole decreases the metabolism, increases the concentration of any drug metabolized by the enzymes. In addition, this potential effect on QT interval increases the risk of the cardiac arrhythmia if used concurrently with other drugs that prolong the QT interval. Antiviral drugs we have discussed in our week 11. So today I am just going to give you an overview of the HIV therapy. So HIV-1 is the most common type of human immunodeficiency virus. It attacks your body's immune system. The virus destroys the CD4 cells. These cells help your body to fight infection. HIV-1 can severely damage your immune system and lead to the acquired immune deficiency syndrome or AIDS. CD4 cells are white blood cells that play an important role in the immune system. Your CD4 cell count gives you indication of the health of your immune system, your body's natural defense system against pathogen, infection and illness. Currently, there is no cure for HIV or AIDS. Once you have the infection, your body can get rid of it. However, there are many medications that control the HIV and prevent the complication. These are known as the antiretroviral therapy or ART. So what are these therapeutic agents which are used for HIV treatment? Which is the first is the reverse transcriptase inhibitor. Reverse transcriptase is an RNA dependent DNA polymerase that utilizes strand of RNA to synthesize double strand viral DNA. This mechanism helps the retroviral gene to get incorporated into the host cell's gene. Various RT inhibitors are developed which on the basis of the binding site and chemical class of the compound are classified into the NRTI, nucleoside reverse transcriptase inhibitor, and the NNRTIs, non-nucleoside reverse transcriptase inhibitors. NNRTI, non-nucleosides 1. So these have the unique antiviral potency, high specificity and low toxicity. NRTIs, these have the nucleoside reverse transcriptase inhibitor inhibit the reverse transcription by causing chain termination after they have incorporated into the viral DNA. This was first group of antiretroviral to be used against HIV. This NRTI lacked 3' hydroxyl group of the deoxyribose moiety. As a result, following incorporation of NRTI, the next incoming deoxyribonucleotide cannot form the next 5' 3' phosphodiester bond needed to extend the DNA chain. Thus, when NRTI is incorporated, viral DNA synthesis is halted. This process known as the chain termination. All NRTI is classified into the competitive substrate inhibitors. We have the protease inhibitor. Protease I have discussed why it is a very important target for the HIV treatment. Because the human protease and the viral protease both have the different structural aspects, right? So it is very important. It is very easy to achieve the selectivity. Protease are the enzyme that cleave the protein molecule into fragments. HIV protease is vital for both viral replication within the cell and release of the matured viral particles. Various protease inhibitors developed for HIV treatment. Some of the FDA approved drugs are the saquanavir, indinavir, atazanavir, ritonavir, nelfiravir, empravir and lopatinib and tirapanavir. Then cholesterol lowering drugs that I have discussed in our last class itself, like statins are the class of the drugs often they are often prescribed by doctors to help to lower the cholesterol levels in the blood. By lowering the levels, it helps to prevent the heart attack and stroke. Studies show that certain people statins reduce the risk of heart attack, stroke and even death from heart disease by 25 to 30%. Statins also known as the HMG-CoA reductase inhibitor. Examples: lovastatin sold under the brand name of Mevacor. It is a compound isolated from the Aspergillus terrace, which was first statin to be marketed. Simvastatin is a brand name is Zocor, which is another lipid lowering drug. Then we have the atorvastatin, which is a lipid-lowering drug, induces the statin class of medication by inhibiting the endogenous production of cholesterol in the liver. Statins lower the abnormal cholesterol of the lipid level and ultimately reduce the risk of the cardiovascular disease. More specifically, statin medication competitively inhibit the enzyme HMG-CoA, which is the hex-hydroxymethylglutaryl coenzyme A reductase, which catalyzes the conversion of HMG-CoA to mevalonic acid. The conversion is critical metabolism aspect for the lipid metabolism and transport, including cholesterol, low density lipoprotein, and very low density lipoprotein. Robustatin this is the structure of the atorvastatin, robustatin, and pitavastatin. Robustatin is used along with the diet to have lower the blood cholesterol of the like and fats and raise the good cholesterol. Pivastatin is a member of the blood cholesterol lowering medication class of statins. Like other statin, it is an inhibitor of HMG-CoA reductase. Then antidepressant drugs. Antidepressants are the class of drug to reduce the symptom of the depressive disorder by correcting the chemical imbalances of the neurotransmitter in the brain. Chemical imbalances may be responsible for the changes in mood and behavior. Neurotransmitters are vital as they are communication link between the nerve cells of our brain. Neurotransmitters reside within the vesicle formed in the nerve cells, which is released by one nerve and taken by other nerve. Neurotransmitters are often taken up by the other nervous system by same levels of release them. This is known as the reuptake. The prevalent neurotransmitter is the brain specific to the depression is the serotonin, dopamine, norepinephrine, noradrenaline. This one. Now, in general, they work by inhibiting the reuptake of the specific neurotransmitter and increasing their level around the nerve cells of the brain. They are such as selective serotonin reuptake inhibitor or SSRI, antidepressant that will affect the serotonin levels of the brain. Monoamine neurotransmitters are the neurotransmitters and neuromodulators that contain one amino group connected to an aromatic ring by two carbon chain like serotonin, dopamine, epinephrine, and norepinephrine. So this is the serotonin, this is dopamine, this is the adrenaline, and this is norepinephrine. Okay. Classification of the antidepressants, like we have the tricyclic and the tetracyclines, monoamine oxidase, and serotonin reuptake inhibitor. So these are the classifications of the antidepressant. When I was discussing about the anti-cancer therapeutics in our last class, which was the yesterday's class, I have discussed about various classes of anti-cancer drugs, but I have not discussed about kinase inhibitors. So kinase inhibitors are newer class of the anti-cancer drugs. Okay, recently there are many kinase inhibitors which are the research is being conducted on the kinase inhibitors as a very potential anti-cancer drugs. Okay. So what is the kinase inhibitor? So first, kinase is the enzyme that catalyzes the transfer of the phosphate groups from high energy phosphate donating molecule to the substrate. Like substrate, when in presence of ATP, kinase, what does it do? It phosphorylates the substrate and makes the ATP the ADP. Okay. So for this phosphorylation, the particular substrate must have an amino acid like serine, threonine, and tyrosine having a hydroxyl group, which gets generally phosphorylated. Okay, that is why the classification of the kinase goes like serine, threonine kinase or tyrosine kinases. Like this one, protein kinases, there are two types, serine threonine kinase or tyrosine kinase. Serine threonine kinase like receptor serine threonine kinases like TGF beta, and non-receptor serine threonine kinase like PK, PKC, MAPK, MAP kinase, aurora kinase. Receptor are just EGFR, PDGFR, FGFR, and non-receptors are like BCR, ABL, and so on. Human genome is like 30 to 35,000 genes are there for the protein kinase gene and protein phosphatase gene. So how does this kinase inhibitors actually work? Okay, and why it is important drug target? So the cell division, the growth factor they attach to the receptors, and this receptor indicates for the cell division. And this now after the receptor, then the kinase this give the signal to the cell, and the cell divide by two cells. So sometimes what happens due to the malfunction of this kinase, sometimes the cancer can happen. Okay, and the kinase inhibitors, how does they work? So basically there is a in case of this enzyme kinase. Okay, so there is a ATP binding pocket where the ATP binds into the particular pocket, and then it actually it start its work by doing this phosphorylation. But this particular ATP binding pocket, if the inhibitor molecules bind in this position, then what will happen? This ATP cannot bind to this pocket, and so the kinase cannot work. So the kinase inhibitor for the design must be like this, that it should fit to the ATP binding site, and it must bind with the hinge region of this kinase inhibitor. So what is a hinge region? So in case of the kinase enzyme, there are two a C lobe and N lobe of this enzyme. One is the C lobe is basically the predominant with the alpha helix sheet, and the N lobe is the beta pleated sheet. Both these lobe are connected by a thin layer of the protein, amino acids, and this is known the peptide chain. This is known as the hinge region. So a compound to be a good kinase inhibitor, it must bind with the hinge region with the hydrogen bonding interaction. So how does it affect? So for example, in case of the BCR-ABL active protein, substrate protein, this is the BCR-ABL kinase. ATP, when it binds, it activates the phosphate and signal for the proliferation and survival, and which leads to the leukemia. But when there is a glyphic is a drug, which is a very good kinase inhibitor drug, when glyphic binds to this in ATP binding pocket of BCR-ABL, what will happen? The substrate no phosphorylation and no leukemia. So that's how this particular kinase inhibitors can work. Okay, so this is not in your syllabus in this detail, but I thought that I should cover up the kinase inhibitor also, because they are very important anti-cancer drugs, and especially in upcoming days, it is many research is going on this kinase inhibitor. So it is good to know about this particular therapeutic also. So this was the last. This was that is all about the summary session. Okay. Okay, so the steps of the drug approval. So I don't know if you have remembered these things, like when we were discussing about the drug discovery. So first is the preclinical discovery, right? So first you have to choose a disease, okay, and which is the unmet medical disease. We also say unmet medical, emergencies, we can say. So whenever there is a particular disease has to be chosen, and for say cancer, like you are going to target the cancer, and then you have to find the target, what target you should choose. So from yesterday I am discussing about the anti-cancer drugs. So there are various enzymes like topoisomerase enzyme, there is a kinase enzyme, there are several enzymes there. So you can choose a particular enzyme. Let's say I am choosing a particular kinase for this particular drug discovery. Okay, then what will happen? First, all the computational studies has to be done to find the heat molecules. Okay, after that, from that heat, the lead molecule has to be discovered. Okay, and then the particular structure activity relationship studies have to be done. Means you have to first synthesize a series of compound, and all the series of compound you have to check that changing which structural activity increases or decreases the particular drug's activity. So this is the structural activities things. After the structural activity relationship studies, when you are getting a particular one or two compounds which is good to go, then that compound is taken to the next step. And this particular compound, what you have to do is you have to try it in an in vitro system on enzymes, or in vitro, then ex vivo, then we go to the in vivo or the animal model. Okay, so there is a mice study, we can do it and many other animal studies can be done. After the animal studies, the compound goes to the, so these all are the preclinical studies. Okay, after a compound passes through all the studies, it goes to the clinical studies. Clinical studies, what so then after the in vivo studies, it goes to the preclinical studies, where the dosing, how much dose of the compound, what is should be the mode of administration, these all are first identified. After that, clinical trials begin. Okay, and a drug is then tested on a humans. So there are different phases of the clinical trial: phase 1, phase 2, and phase 3. First, small group of people are tested. The drug is tested on small group of people, followed by the larger group of people. Then in the phase 3, more people are taken into the clinical trials. After the phase 3, there is a FDA approval, or in case of US, you have to take the FDA approval. After the drug passes to the FDA approval, it goes to the market. Okay, and after that, there is a phase 4 clinical trial also, which is known as the post market surveillance, because whenever you are doing the clinical trials on a selected number of people, but when the drug goes to the market, what happens? This larger number of people are using the drug, so some kind of side effect may rise, or some kind of inactivation may occur. Okay, so this is dealt with the that post-market surveillance. Okay, so these are the various steps of the drug discovery. If you go to the week 9 of our course content, you will find the details of the drug discovery steps. Okay, there I have mentioned and I have also given you all the data. So so as I have mentioned, when to I know hope you know all when to where to find. So let me just share my email ID because this is the last class. So if you have any doubt anymore, please let me know via email. Okay. So you just have to go to the this portal of the SWAM. Then you will find there is the at the left panel at the end you will find the problem solving session. If you click on that portion, you will find that problem solving session recording. If you click, you will get all the class recordings present here in this page along with the PPTs. Okay, it will contain a Google Drive link which will lead to a Google Drive like this. This drive contain all the group all the PPTs which I have discussed till now. All the class materials is present. You can easily download and you can read from here. Okay, so thank you all for joining the class. I hope it was helpful for you for understanding various aspects of organic chemistry and its application in biology and drug development. Today it was our last class. Thank you all for joining. If you have any problem or any doubt or any query, please feel free to reach me through the email. Also, you can put your queries via the discussion forum of the organic chemistry in biology and drug development. Okay. Thank you all. I will close the session for today. If there is no doubt. Okay, so I am closing the session for today. Thank you for joining. Have a good day.